we finally welcome William Botta from the New Jersey Institute of Technology in the US, who is going to talk about the complex interplay of sensory system that is affecting mass behavior in zebrafish.
Hello, my name is William Botta.
Sorry for that. We'll have to wait for William to be with us. William, are you
I'm
here?
here.
Oh, you're here. Okay, great. If I'm not wrong, like last time that I finished the conference, it was already you, the last talk of the conference, right?
Oh yeah, it was last time too.
Yeah. So like it's getting a tradition. So like I will start the talk and then we will come back for the question, William.
Sounds good.
Hello, my name is William Botta, and today I will be talking to you about the complex interplay of sensory systems affecting mass behavior in zebrafish. I will be starting by defining what mass behavior is. I am defining it as coordinated behavior during which participants adopt similar conduct. Think of swarming behavior in insects or schooling in fish, or collective decision making, such as a group of buffalo stampeding across a plains in order to find a new location, or mass avoidance behavior, such as this group of fish avoiding this predator in the top right. A group in this way can accomplish more goals effectively than any individual. If one individual in the group finds food, then all of a sudden everybody can navigate to that food together. If one of them sees a predator and escapes, the rest will follow suit and avoid that predator. And living in a group like this, if one of them starts mating with another, that's a cue for many others to know that it's safe enough to mate themselves. However, many organisms follow rules in order to organize, and these rules can break down, which leads to negative outcomes, such as stock market crashes, or tramplings due to misappropriated stampedes, or this death spiraling behavior that we see in ants. My research focuses on the factors that control the initiation, propagation, and extinction of these mass behaviors. And we know that there are many factors that can influence collective behavior, such as environmental factors, the presence or absence of obstacles such as those on the left, or perception, such as the fish seeing the other in the right, which can lead to an adoption of behavior, or mental state, wherein, depending on the mental state, that may change the likelihood of behavior being adopted by an individual. Today, I will be focusing on perception. And when I do my research, I use larval zebrafish as a model. There are many benefits to this, such as a well-studied behavioral repertoire. At this age, they exhibit more limited behaviors and do not shoal together. They are transgenically accessible, so we can manipulate their behaviors through different transgenic lines or label different neurons to see how they affect their behavior. They're easy to grow and experiment on. We can get a large number of fish relatively quickly, and they exhibit their behaviors at a very young age. And they possess specific sensory organs that are relevant to the behavior, such as eyes for vision and a lateral line system that allows them to sense local changes in water pressure. Another benefit to using zebrafish is that they exhibit mass behavior. Groups of zebrafish in a controlled area will exhibit these spontaneous collective circling events, such as the one that you're seeing on the right, where a wave of behavior will travel around the outside of the arena. And previous research has shown that as density of this group increases, the likelihood of exhibiting that response increases as well. My initial experiments were looking at effects of lighting and arena size and therefore group size on this behavior. So we use this behavior rig on the left, which consists of a high-speed camera in a dark box with a projector and an infrared light, allowing us to project different light levels and projections to the fish to exhibit behaviors, as well as to allow us to record them while they are in darkness. We use custom-made arenas, such as the one in the center and on the right, that allow us to control the water level and the amount of area that they have, and therefore we can control the group density and look solely at effect of group size while maintaining a constant density using an arena such as the one in the center with 50 fish. That is a quarter of the area of the one on the right with 200 fish. We then take 10-minute long videos with this setup and use optical flow in order to process how the fish are swimming to determine when collective circling events are happening. We use pixel measurements between each image in the video to measure, to calculate motion within the arena, which creates an image like the one in the center, which shows areas of high and low motion between two images, and then we can take the numerical data from that image in order to calculate how much motion is occurring, such as in the graph on the right, which shows time in the x-axis and motion intensity on the y-axis. And we can see that there are periods of high motion that correspond with the circling events, and then periods of low motion where they are not circling. So when I take all of these together, so far what we have found is that collective circling is more likely to occur in the dark. On the left graph, we see group size being graphed on the x-axis and number of circling events on the y-axis. And what we see here is, so far, fish in the dark are more likely to exhibit collective circling events than those in the light, where it was higher in all of them, with the light being 0 for 50, or very close to, the number of circling events being close to 0 in the light for 50, 100, and then 150. And then specifically, once we reached 200 fish in that larger dish, we saw a marked increase in number of circling events in both the light and as well as in the dark. And this has tested to be significant using a generalized linear mixed model. And then on the right graph, we have group size again on the x-axis, but this time average circling event duration on the y-axis. And we see here that those in the dark tended to be relatively consistent in terms of duration. Just a slight spike in those in the light at the 200 level, and this is due to an artifact of not having many, if any, circling events in the light videos. This was determined to be not significant, so the circling event durations tend to be pretty consistent. In addition to looking at effects of light, we have also looked at the lateral line. One of the undergrads working with me, Nikki Anagnosto, initially wanted to determine that we could use gentamicin, which is an established antibiotic, to affect the lateral line system. And so she bathed the lateral line in 20 micromolar gentamicin sulfate for 24 hours in order to ablate the lateral line, and then rinsed those fish off in order to confirm that the fish were, that the lateral line was ablated. She anesthetized them with MS-222, which is an anesthetic, and then took pictures, and then took those fish and stained them with DASPI, which labels neuromasts, for 15 minutes, gave them another rinse, and then took more pictures after anesthetizing them a second time. And we got pictures that look like this. The fish on the top is a control fish that was not bathed in gentamicin, but was instead bathed in fish water. And then the fish on the bottom has been bathed in the gentamicin and had the lateral line ablated. And you can clearly see the neuromasts in the control fish. Those are those green spots. And then they are not being seen in the gentamicin fish. And when she counted them out on a number of fish, she found that the gentamicin ultimately was very good at ablating the neuromasts on the lateral line, where we didn't see any of the neuromasts, whereas we did see them in the controls. She followed this up by doing group experiments with lateral line ablated fish. So she took 50 fish and either ablated the lateral lines using the 20 micromolar gentamicin or used controls, bathing them in fish water for 24 hours, and gave them a rinse, and then conducted the 10-minute long behavior trials just as before, while taking some of them as a subset and staining them with DASP in order to make sure that the neuromasts were still being ablated in those individuals. And what she found is that the ablation of the lateral line completely shuts down this circling behavior. So in this graph, on the x-axis, we have our treatment, with control being on the left and the lateral line being on the right, and then the number of circling events again being on the y-axis. What we see here is just as before. In the control fish, the fish in the dark exhibited higher levels of circling events in 10 minutes than the ones in the light, whereas when the lateral line was ablated, it completely shut it down in both the dark and the light. No collective circling events were observed. So I took this and decided to ask a second question, which is regeneration of the lateral line. Zebrafish larvae at this age exhibit a miraculous ability to regenerate many of their limbs and other relevant sensory structures, such as the lateral line. And so I wanted to test, number one, whether they would be able to regenerate after 48 hours. And so similar to before, and number two, whether this group behavior continued after the lateral line was lost and then regained. And so similar to Nikki's group experiment, I took 50 fish and bathed them in gentamicin for 24 hours or fish water as a sham, and then I rinsed them out and then gave them a behavior analysis while setting aside a subset for imaging to make sure that it worked. And then after 48 hours, I put them back into the behavior chamber to film them again while also imaging some more and making sure that the lateral line had come back. And I did see that the lateral line had come back. And then in terms of the group experiments, I saw that similarly to before, the control fish exhibited collective circling both on the day of ablations or sham ablations, and then 48 hours later they still continued to exhibit these collective circling behaviors. And then in the lateral line ablated fish, we saw that they lost their ability to do this collective circling behavior, and then 48 hours later, however, when the lateral line had recovered, they continued doing the circling behavior. This is N of 3 data, so I have not been able to do some stats on it yet. I still want to collect some more trials of this, but the trend shows pretty significant ability of this to be recovered. So in conclusion, we know that absence of light increases this circling behavior. The lateral line is required for this circling behavior to be expressed, and if it is ablated, we see pretty clearly that it comes back after recovery. This suggests that there is an interplay between light perception and lateral line controlling the behavior, where the lateral line has to be there and then the light perception is modulating it. In future directions, I wish to test sense of touch on this as well, another behavior that may be important for this, another perception that may be important for this behavior. I have another undergrad that I am working with on a similar project who has presented a poster earlier this week. I highly recommend you all go check that one out. Bansari has done great work looking at the effects of mental state with some preliminary data on caffeine and its effect on this, and we want to test effects of fluoxetine as well to see if a heightened anxious mental state or a calm mental state may affect the ability of them to do this behavior. And I also want to analyze collision rates in the light and the dark in order to see if it's more a matter of them colliding with each other that is causing this behavior. At this point, I would like to acknowledge my funding sources, such as the Animal Behavior Society, which has given us a grant towards working on this behavior, as well as the New Jersey Institute of Technology, which pays for a lot of my travel funds. I would also like to take this time to thank the Swarm Lab for their continued support in analysis of collective behavior, as well as the Severi Lab for their expertise in zebrafish and neurobiology and their assistance on this project. Thank you so much, and I hope you have a great day.
Thank you, William. Are you with me?
Yes, I am.
Yeah, great. Thanks for the presentation. So like I have some question myself to start. Like, I mean, if I correct me if I'm wrong, but like I think the gentamicin sulfate is not like fully selective. There's still a risk that some other cells are impacted, right?
There is some risk, but at the low concentration that we're using over that period, it's really only going to affect surface level cells. And because at that age, the neuromasts of the lateral line are right on the outside, it shouldn't be doing too much damage.
Mm-hmm. And I was
We also,
yeah, no,
I was
go ahead.
just going to say, anecdotally, when we observe the fish in the videos from the gentamicin, we see that they can swim normally.
Okay, yeah. I mean, I was actually about to ask whether like you have tested your lateral line altered strain, if you have tested them with the light dark transition test in order to see whether like locomotion wise they were still the same.
Right. So we haven't done kinematics testing specifically. I can only say that when we watch them, they do swim as if they're not affected by it.
Mm-hmm.
In terms of giving them a transition test, I have not done that.
Okay. Okay. And so one last question is that I was wondering if you were planning in the future to maybe actually make the same type of test, maybe running some calcium imaging using some mutant that could allow you to see activation of the lateral line or something like this.
We would absolutely love to. We have a grant in the works getting us a microscope that would allow us to do the calcium imaging of the entire group collectively.
Yeah.
Um,
Super cool.
but yeah.
Okay, I will stay tuned then. We also have received some question on the chat. So like we have AB, we say great talk. I was wondering like about the group size effect, especially in the light condition. There seems to be some thresholding effect. Is this density dependent? Have you tried changing arena area?
Okay, so in terms of looking at density, for my experiments, all of the arenas were conducted at the same density. Previous paper put out by our lab by Hader Zaki was they were the one who initially pioneered the behavior. Looking at this behavior, and they did a number of density tests, starting with a very low density and ramping up to about 130 fish in the dish that of the same size that we used, which is why I used that size of dish. And they found that it's definitely a density dependent behavior. But it was interesting to see that it also seems to be numerically modulated as well.
Okay, thanks for the answer. We have received question from Bastormenta Forever. How do you safely transfer the fish from the water to the trandomycin and back into fresh water?
Sorry, could you repeat that?
How do you safely transfer the fish from the water to the gentamicin sulfate and back into fresh water?
Right. So we actually use transfer pipettes. Um, and we can transfer them. They're really small, and so you can just suck them up into the transfer pipette, and we put them right into a petri dish, and then we add a large amount of that molar concentration solution.
Oh, thanks. And so like finally, we have a question from Tang who say exciting. Is the behavior after lateral line regeneration the same as before ablation, or is there any reduction in performance?
So far, I'm not entirely sure on in terms of like the actual like behavior. I haven't done kinematics analysis. Based on the results that I was seeing, it seems like at least they are running circling events slightly less frequently.
Mhm.
But that's also just, we don't have enough replicates for me to confidently state that yet.
Okay. And like, I mean, I was wondering also, like, do you plan at some point also maybe to move forward in adult and see like if it's something that is conserved along development, or like you will just for now focus on larvae?
Right. For now, I'm definitely focusing on the larvae. There's a lot to do in the system, but it would be so cool to look at how this develops, especially once they transition into being more social and
More
shoaling.
social, yeah. Exactly. Okay, very interesting. Yeah, okay, but like I think we don't have more questions. So like I guess that if people have more questions to ask, do not hesitate to go on Discord, and William, I'm sure like will be happy to reply to your question on Discord.